Students, today we will be discussing a new topic. It is the axis specification in Drosophila. Whenever we speak of axis specification, we know that we are talking about the formation of some features in certain orientation, in certain direction. So we have the anterior posterior axis, we have proximal distal axis, and we have dorsal and ventral axis. These axes you have studied when I was discussing about the limb development in tetrapod. So in Drosophila also, when the egg starts its developmental journey to become an adult, it is almost spherical, or here in the case of Drosophila it seems to be oval type. So from this stage it starts its journey to become an adult Drosophila. So the axis must be specified. It should be known of this egg which part will form the head region, which part will form the thoracic region, and which part will form the abdominal region. We all know that insect's body is divided into head, thorax, and abdomen. So it has to be decided, there has to be certain signal which must be given to this egg so that it forms the anterior part, the posterior part, and the details in these part. Anterior region, we have head, or we also have thorax, and then posteriorly we have the abdomen. These structures are also segmented because we know that arthropods are segmented. Segmentation is one of the characteristic of arthropods. They have true segmentation. So these axis must be formed so that we know which part will form the head region and which part will form the tail or the abdominal region. Then there must be some signals which should tell the head region to form the eyes, antenna, then the mouth parts. Then there should be certain signals which should tell the thoracic region to form the wings, to form the legs. And similarly, there are details in each segment which must be specified. So, about the insect egg, we know that most insect eggs undergo superficial cleavage. That is, once one cell divides, its nucleus divide, the division of nucleus is fast as compared to division of the cytoplasm. Therefore, what we get? We get a syncytial blastoderm, which means that all the cleavage nuclei are present within the common cytoplasm, and no cell membrane is present for the nuclei so as to distinguish them or demarcate them. This type of division is found in insect, and this division or cleavage is called the superficial cleavage. So we are not discussing about the cleavage. Our focus is on anterior and posterior axis specification in Drosophila. So in the picture you can see Drosophila, anterior region comprising of the head, thoracic region and the posterior region. जब हम एंटीरियर पोस्टीरियर एक्सिस की बात करते हैं, उनकी विशिष्टता की बात करते हैं, तो एंटीरियर यानी अग्र भाग में हमें पता है कि हेड रीजन होता है, थोरेसिक रीजन होता है, और जो पश्च भाग है यानी कि द पोस्टीरियर एंड, इट कंप्राइजेज ऑफ द एब्डोमेन. ये इंसेक्ट की हम बात कर रहे हैं. तो ये स्पेसिफिकेशन किस प्रकार से होता है, मैंने अभी आपको एग दिखाया था, ये ड्रोसोफिला का एग है। इसके अंदर ये निर्धारित करना बहुत ज़रूरी है कि इस गोलाकार स्ट्रक्चर का या ओवल स्ट्रक्चर में सिर कौन से भाग से बनेगा, थोरैक्स कौन सा भाग बनाएगा और एब्डोमेन कौन सा भाग बनाएगा। और उसके बाद ये निश्चित होना भी आवश्यक है कि इन सभी सेगमेंट के जो स्पेसिफिक स्ट्रक्चर्स हैं वो कौन से भाग बनाएंगे। तो